So now we're going to talk about penicillin. I'm going to split this into two different talks because it's a lot of material. We won't, I won't cover as much in other drugs, but for this particular class, I'm going to go more in depth on it. So I'm coming back to this slide that I talked about at the end of the last section. I just wanted to make you familiar with a term that gets used, and the term is four-quadrant coverage. And what that means, typically when we have an animal come in the clinic and they have an infection and we want to give them something right away, we want to get started on treating that infection. We may send out a sample for culture and sensitivity, but in the meantime, we're going to put them onto a particular drug. This is called empiric treatment. And I'll have a slide later where I'll kind of bring it up again and show you the word. But um, sometimes if we, depending on what we know about the type of bacteria that may be causing the infection, we may think it may be a couple of types of bacteria, or we don't know, and we want to make sure that we get onto this get a handle on this infection right away. So we might just go ahead and do what's called four-quadrant coverage. And that is, these are the four quadrants. We're going to make sure that the drug or drugs that we put the animal on targets aerobic bacteria, anaerobic bacteria, gram-positive, and gram-negative bacteria. That means that we've kind of covered every possibility. So it's not the most ideal way to do it unless we know that we do have these two different types, or have bacteria that are following these classifications, all of these classifications. It's better to use one drug and target um, what is best there, what you want to treat. And that's best done with culture and sensitivity, but it takes several days to get the results back from culture and sensitivity, and it costs the owner some money. So sometimes we're not able to do that. And so that term, four-quadrant coverage, I will bring in a chart that is supplied by a drug company that helps veterinarians select antibiotics. And it kind of goes over that. It shows the four quadrants. It puts them in a little square, and it kind of helps in the selection and based on these classifications. All right, so penicillins, they are still very commonly used. There is a considerable amount of resistance to these drugs, but we still do use them, and we've done things to them to make them more useful in situations where they would have otherwise been not so useful because of resistance. Excuse me. So there's a variety of types here, and there's the natural penicillins or penicillin G. This is still derived from growing the fungus, and most of the rest of these are synthetically derived. And then there's broad spectrum aminopenicillins, and that includes ampicillin and amoxicillin. Broad spectrum refers to what I just was talking about. They covers a variety of gram-negative, gram-positive, aerobic, anaerobic. It's more broad spectrum. There are penicillinase-resistant penicillins, and I'll talk about penicillinase soon. Extended spectrum penicillins. So again, just kind of named on what they target. And then we have potentiated penicillins. So penicillins work on the cell wall of the bacteria, and I showed you that video of the bacteria rupturing, complete with sound. And the big thing that you have to know about it is we have to have actively dividing um, bacterial colonies in order for penicillin to have an effect, because it acts on the actual construction, one of the enzymes that is used to construct the cell wall. And without that enzyme, the cell wall is not strong, and so it will rupture. The reason why that's important is I'll talk about it again here in a minute. Generally, penicillins are effective against gram-positive bacteria. Not as much, but some gram-negative bacteria may be affected as well. Again, remember this just has to do with, it's a characteristic, when we talk about gram, it's a characteristic of a stain to be taken up by the outer layer in a bacterial cell wall. But that ability to take up the stain or not also indicates an ability for the antibiotic to get into the cell, to that cell. And penicillins are effective against a spectrum of aerobic and anaerobic bacteria. So I talked about a couple of classifications in the last slide. Broad or extended spectrum just means that they have an extra area that they may extend, that particular group has an effect on. So this is that word empiric therapy, and that just is the term that we use to describe when we select antibiotics based on what we know, what we see in the patient, what we know of these types of typical infections, and we're starting therapy without a culture and sensitivity test. We are just using our knowledge of what's typical for different types of infection and selecting antibiotics that way. And that can be revised after, if you do a culture and sensitivity. I know I've had this happen to myself where I've been called and I've been told, you know what, that is not going to work on that infection, so we're going to switch you to a different antibiotic. So it's not set in stone once you start a course of antibiotics. So the reason why you need to be aware of the actively dividing colonies for penicillin to be effective is that sometimes if we do start an animal on a couple of different types of antibiotics and we put them on a static type of antibiotic that's going to inhibit cell replication, then if they have a penicillin on board, that's not going to be effective. You should not then give the penicillin because you don't have an actively dividing colony. So it's just something to be aware of, in particular with penicillins and also cephalosporins that rely on the bacteria to be actively growing. You don't want to use it together with a drug such as doxycycline or doxycycline, which is a tetracycline, together with an amoxicillin, because doxycycline is going to stop the replication of the bacteria, and so having penicillin in the same infection, it's going to be useless. But I want to emphasize that you can use a bactericidal and a bacteriostatic drug together. Just don't use a cidal that depends on the bacteria to be actively growing if you use it with a static drug. So there's just a couple that are in this area, and it's pretty much penicillin and cephalosporin, but it's just something to be aware of. Okay, so in general, penicillins are pretty well absorbed when we inject them and when we give them orally, except for penicillin. So penicillin G is the one. It's inactivated by gastric acid. Gastric acid begins with a G, so hopefully that'll help you remember it, that we can't give penicillin G orally. We give it injectable. And some bioavailability is reduced when we give oral penicillins with food. So you want to be aware of that. And generally, you don't want owners to be giving it with food. But amoxicillin, which is a really common one that we use, we can give that one with food. Penicillins are distributed pretty well in the body. We're going to talk about a couple of areas, that are hard to get antibiotics into. and they generally have to be lipophilic in order to get into them. So brain, eye, I'm not going to talk about it here, but brain and eye are the main ones here. The globe of the eye can be tricky to get into, joints, and the prostate gland. Those are all difficult areas, and the central nervous system. So penicillins are typically not used for central nervous system infections because of that reason. They don't tend to get into there. It's metabolized in the liver, or they are, as a class, metabolized in the liver. The excretion is really important because penicillins are well excreted via the kidneys. Because of this, they tend to collect in high concentrations in the kidneys and the urinary bladder. And so they can be used to treat infections there because they tend to go there and have high concentrations. So these are good choices typically for kidney and bladder infections.